Bismillah ar-Rahman ar-Rahim. This is the second and the last lecture in the chemistry of carbohydrates. The objectives of this lecture are to study the structure of disaccharides, oligosaccharides and polysaccharides, including the homopolysaccharides and heteropolysaccharides, and also to describe the structure of mucopolysaccharides, the glycosaminoglycans, and also to describe the structure of glycoproteins and proteoglycans. Disaccharides are made up of two molecules of monosaccharides, whether from the same type of monosaccharide or from different monosaccharide units. These monosaccharide units within the disaccharide are joined by O-glycosidic bond. The general chemical formula for disaccharides is C12H22O11. The most important disaccharides are maltose, lactose, and sucrose. Other examples of disaccharides are cellulose and 3-halose. We start with the disaccharide maltose. It consists of two glucose molecules linked by alpha 1,4-O glycosidic bond. Why it is 1,4? Because it's formed by the linking between carbon number 1 in the first glucose molecules with the carbon number 4 in the second glucose molecule. Therefore it is 1,4. Why it is alpha? Because the hydroxyl group in the anomeric carbon of the first glucose molecule lies below the ring. Therefore it is alpha 1,4. And maltose, it is written as alpha D glucopyrenosyl 1,2,4 alpha D glucopyranose. Why? Because the hydroxyl group in the anomeric carbon of the two glucose molecules in the maltose lies in the alpha or occur in the alpha configuration. In other words, lies below the ring. Maltose, it is an intermediate in starch hydrolysis by the action of the enzyme amylase. Maltose is a reducing sugar. It gives a positive result with the Benedict reagent. Why? Because the second glucose molecule have a free carbonyl group, and for this reason maltose is a reducing sugar. The other disaccharide is the lactose. It is formed by reaction between the galactose and the glucose molecule, and the O-glycosidic bond between glucose and galactose molecules in the lactose is in the form of beta 1,4. 1,4 because it occurs between the anomeric carbon of galactose with the carbon number 4 in the glucose. Therefore it is 1,4. Why it is beta? Because the hydroxyl group in the anomeric carbon of galactose lies above the ring. Therefore it is in the beta configuration. Therefore it is beta 1,4. And lactose, it is written as beta D galactopyrenosyl 1,2,4 beta D glucopyranose, because the hydroxyl group in the anomeric carbon in both galactose and glucose lies above the ring. Lactose, it is a milk sugar. It is a reducing sugar. It gives a positive result with Benedict's reagent. Why? Because it contains a free carbonyl group in the anomeric carbon of the second molecules of lactose, that's the glucose. The other disaccharide, the sucrose, it is made up of one molecule of glucose and one molecule of fructose linked by alpha 1 beta 2 or glycosidic bond. This is the 1 to 2 or glycosidic bond between the carbon number 1 in glucose with the carbon number 2 in fructose, and it is called alpha 1 beta 2 because of the configuration of the hydroxyl group in the glucose and fructose residues. Here it is important to mention that the fructose molecule is inverted by 180 degrees in order to bind with the molecules of glucose for the formation of sucrose. Sucrose, it is also called table sugar or cane sugar. It is a non-reducing because it contains no free carbonyl group, and it is written as alpha D glucopyrenosyl 1 to 2 beta D fructofuranoside. Regarding oligosaccharides, they are carbohydrates consist of 3 to 10 molecules of monosaccharides. They are attached covalently to the outer surface of proteins of cell membrane, forming glycoproteins which serve as receptors. Also, many antibodies and coagulation factors, they contain oligosaccharides unit. Examples of oligosaccharides include maltose, consists of 3 molecules of glucose linked by alpha 1,4 or glycosidic bond. Other example of oligosaccharide is the raffinose, which consists of 3 molecules, 1 galactose, 1 glucose, and 1 fructose. Regarding polysaccharides, they are more complex carbohydrates. They yield or result in more than 10 molecules of monosaccharides on their hydrolysis. These polysaccharides can be further subdivided depend on the type of monosaccharide they are derived from. They can be divided into 2 types. The homopolysaccharides, they are derived from the same type of monosaccharide. In other words, they are polymer of the same monosaccharide units, or they contain the same type of monosaccharide. For example, starch, it is a polymer of glucose. It is of a plant origin. Glycogen, it is also a polymer of glucose. It is of animal origin. Both starch and glycogen, they contain glucose molecule linked by alpha 1,4 glycosidic bonds with branching point at alpha 1,6. It is important to mention that glycogen, it is a highly branched molecule than starch. Inulin, it is also a homopolysaccharide. It is a fructose polymer. Cellulose, it is a glucose polymer, but the type of glycosidic bond is different. It is a beta glycosidic bond. Chitin, it is also a homopolysaccharide containing the same monosaccharide unit. That's the N-acetyl glucosamine. It is contain only N-acetyl glucosamine linked by beta 1,4 or glycosidic bond. While heteropolysaccharides, they are polymer of different molecules of monosaccharides or their derivatives. In other words, the heteropolysaccharides are polysaccharides that contain different types of monosaccharides. For example, hyaluronic acid, chondroitin, sulfates, heparin, heparan sulfate, keratin sulfates 1 and 2, and dharmatan sulfate. Now we discuss the homopolysaccharides. The polysaccharides that are consist of the same building block or the same monosaccharide unit. Starch Starch, it is a polymer of alpha-D glucose. Starch, it is the most important dietary carbohydrate of plant origin, present in cereals, potatoes, and other vegetables. Chemically, starch consists of two types of glucose polymers. The non-branched one, the amylose, and the branched one, the amylopectin. The non-branched part of starch, which constitute around 15 to 20% of starch, it is a non-branched structure made up entirely of alpha-D glucose. These glucose molecules linked by alpha-1,4-O glycosidic bond, so it is head-to-tail polymer of around 4,000 glucose molecules. This is the structure of the amylose, which constitute around 15 to 20% of the starch molecule. It is a helical structure. This is the alpha-1,4, the linkage between carbon number 1 of the anomeric carbon of this glucose molecule with the carbon number 4 of the second glucose, and it is called alpha-1,4 because the hydroxyl group here lies below the ring. The vast majority of starch consists of the branched one, the amylopectin, which constitutes around 80 to 85% of the starch. It is a highly branched chain composed of alpha-D glucose units united by or linked by alpha-1,4-O glycosidic bond, while there is a branching point at the alpha-1,6 linkage, and the branching points occur at 1 in every 24 to 30 glucose molecules in the carbohydrate chain, and therefore starch, it is called a heterogeneous homopolysaccharide. It is a homopolysaccharide. Why? Because it consists of the same molecule of monosaccharide. It contains only glucose molecule. Why it is heterogeneous? Because it is a combination of non-branched part, which is the amylose, and the branched part, which is the amylopectin. Therefore, it is called a heterogeneous homopolysaccharide. This is the amylopectin molecule. This is the chain of glucose molecules in the amylopectin. This is the glucose linked to the second glucose by alpha-1,4, and here there is a branching point which occur 1 at every 24 to 30 glucose molecule, and the branch occur at the alpha-1,6. Here it is more clear, where this is the alpha-1,4, this is the alpha-1,4. This is also, it is more clear, and this is the alpha-1,6. This is the branching point in the carbon number 6 in the ring, in the glucopyranose ring. The second homopolysaccharide is glycogen. It is the storage form of polysaccharide in animals, and it is sometimes called the animal starch. It is a highly branched molecule like amylopectin, composed only of alpha-D glucose molecules, also linked by alpha-1,4-O glycosidic bonds, with the branching points occur at the alpha-1,6 glycosidic bonds. However, the difference between glycogen and amylopectin is that the glycogen molecule is highly branched. Therefore, the branching point occur at a shorter chain of glucose molecules, where the branching occur 1 at every 12 to 14 glucose molecules, while with amylopectin it is a little bit longer, occurs at, as I said earlier, 1 in every 24 to 30 glucose molecules. This is the molecule of glycogen. It is highly branched. This is the glucose molecules in the glycogen. There is alpha 1,4 glycosidic bond, while the link the branching point occur at alpha 1,6. This is the alpha 1,4 and this is the alpha 1,6. This is the alpha 1,6. This is another picture showing the molecule of glycogen for more clarification. This is the alpha 1,4 or glycosidic bond, and this is the branching point, occur at every 12 to 14 molecules of glucose, occur at alpha 1,6. During meals, any excess of glucose is stored in the body, usually in the liver and also in the muscles, in the form of glycogen by the process called glycogenesis. Glycogenesis is the process by which the glucose is deposited in the form of glycogen. While between meals during fasting state, the glycogen is broken down in order to release glucose by the process called glycogenolysis, the process of breakdown of glycogen. The third homopolysaccharide is the inulin. Inulin, it is a fructose polymer. It is a homopolysaccharide made up of the fructose molecules. It has no dietary importance in humans. It has a low molecular weight and it is readily water soluble. It is used to determine the glomerular filtration rate, GFR, in the assessment of renal function. This is the molecule of inulin. It is a fructose polymer. The fourth homopolysaccharide is a cellulose. This is structural polysaccharide. It is the main constituent of plant cell wall. Chemically, it is unbranched homopolysaccharide consisting only of glucose residues but of the beta configuration, and they are linked by beta 1,4 linkage, forming long, straight and branched chain. These carbohydrate chain, they are strengthened by cross-linking hydrogen bonding, forming a structure called the fibrils. The cellulose, it has no importance in human because the human gut does not contain the enzyme which is responsible for the hydrolysis of beta 1,4 or glycosidic bond. Therefore, cellulose cannot be digested in our body. This is the molecule of cellulose. This is the carbohydrate chain consisting of beta glucose residues linked in the beta 1,4. Why beta 1,4? Because the hydroxyl group in the anomeric carbon of this glucose lies above the ring. This diagram shows different homopolysaccharide. This is the starch molecule. It consists of two portions. This is the linear or the non-branched part of starch, the amylose, and this is the branched portion of starch, the amylopectin, where this is the glucose linked in alpha 1,4 and this is the branching point at alpha 1,6. This is the glycogen molecule, which is a highly branched molecule, more than that of amylopectin, and this is the structure of cellulose. These are the cellulose molecule, and this is the fibrils structure. Why? Because the cellulose, the glucose molecules within the cellulose are tightened by the hydrogen bonding giving this structure called the fibrils. As I said, the human gut contains no enzyme that digests the beta 1,4-O glycosidic bond. Therefore, the cellulose cannot be digested, and therefore it is part of the insoluble fibers that may aid in colonic function. Regarding the homopolysaccharide chitin, it is a structural polysaccharide present in the external skeletons of crustaceans, al-qashriyat, and also present in insects and also in mushrooms. It consists of N-acetyl D-glucosamine, these joined by beta 1,4 glycosidic bonds. Now we discuss the heteropolysaccharide. These are the polysaccharide polymer consisting of different units of monosaccharides or their derivatives. The mucopolysaccharides or the glycosaminoglycans, these are types of heteropolysaccharides, consist of different monosaccharides units or their derivatives. But what is particular regarding glycosaminoglycans is that they are consist of amino sugar and uronic acids. Therefore, the mucopolysaccharides or the glycosaminoglycans are these heteropolysaccharides consisting of repeated units of amino sugar and uronic acids. Example of glycosaminoglycans include hyaluronic acid, chondroitin sulfate, keratin sulfates 1 and 2, heparin, heparan sulfate, and dharmatan sulfate. These glycosaminoglycans may be attached to a protein core, forming proteoglycans. Regarding glycosaminoglycans, a glycosaminoglycan is an unbranched polysaccharide made of repeating disaccharide units. One component of the disaccharide unit is always an amino sugar, which is either D-glucosamine or D-galactosamine. The second component of the repeating disaccharide unit is a uronic acid, either L-glucuronic acid or L-idiuronic acid, with the exception of the homopolysaccharide keratin sulfate, which contain no uronic acid, but instead it contains galactose. Also, all types of glycosaminoglycans or mucopolysaccharides are sulfated. They contain sulfate, with the exception of hyaluronic acid. Therefore, hyaluronic acid, it is the only unsulfated glycosaminoglycan. It is the only glycosaminoglycan that have no sulfate. Also, a glycosaminoglycan may contain sulfate group either as an ester or as an sulfate. For example, in heparin sulfate and in heparin. The hyaluronic acid, it is, as I said, it is non-sulfated mucopolysaccharide. It is present in a variety of tissues, including the synovial fluid, in the vitreous body, in the eye, in the cartilage, and also in loose connective tissues. It is also present in bacteria. Chemically, the hyaluronic acid consists of an unbranched chain of repeating disaccharide units containing D-glucuronic acid, this is the D-glucuronic acid, and the N-acetylglucosamine. These two compounds are joined by beta 1,3-O glycosidic bond. and each disaccharide, this is the disaccharide unit, each disaccharide is attached with the next disaccharide via beta 1,4 or glycosidic linkage. This is another figure showing the structure of hyaluronic acid. The enzyme hyaluronidase catalyzes the hydrolysis of beta 1,4 or glycosidic bonds of hyaluronic acid, and this results in reduction of the viscosity of the compound. The enzyme hyaluronidase present in the testicular secretion therefore aid in dissolving of the substance surrounding the ova, therefore it facilitates in the penetration of the ova. Also, it is secreted by some pathogenic bacteria, facilitating their diffusion and spread into the tissues. The second glycosaminoglycan is the chondroitin sulfates. We have chondroitin 4 sulfate and chondroitin 6 sulfate. The chondroitin consists of an branch chain of repeating disaccharide units containing also the glucuronic acid, but here there is an acetyl galactosamine instead of the unacetyl D-glucosamine, which is present in the hyaluronic acid. They are these two components linked by beta 1,3 or glycosidic bond. This is the beta 1,3 or glycosidic bond, and each disaccharide is attached to the next disaccharide unit by beta 1,4. As I said, there is sulfated derivatives, the chondroitin 4 sulfate and chondroitin 6 sulfate. These sulfated compounds are the major components of cell wall, cartilage, bones, cornea, and connective tissues. This is also another description of the structure of chondroitin sulfate, whether the 4 or the 6. Now, heparin is a mucopolysaccharide which is highly sulfated compound. It is produced by the liver. It is a polymer of repeated units of uronic acid and unsulfated glucosamine. The uronic acid could be L-uronic acid or the glucuronic acid. However, 90% of the uronic acid that is present in the molecule of heparin is of the L-uronic acid. Functionally, heparin inhibits the formation of thrombin from prothrombin, therefore it is used as an anticoagulant. The heparan sulfate, it is an extracellular glycosaminoglycan. It is present in many cell surfaces as a proteoglycan. What are the differences between heparan sulfate and heparin? Heparan sulfate contains the uronic acid, glucuronic acid, instead of the iduronic acid that is present in heparin. Therefore, heparin contains mostly the uronic acid as a glucuronic acid, while the uronic acid in heparin is the iduronic acid. The second difference is that heparin contains fewer unsulfates than heparin. Concerning keratin sulfates 1 and 2, they consist of repeating disaccharide units. The disaccharide unit in keratin sulfates 1 and 2 consists of N-acetyl glucosamine. This is the N-acetyl glucosamine and galactose. It is important to say here that the keratin sulfates 1 and 2, they are the only glycosaminoglycans that have no uronic acid. There is no uronic acid in keratin sulfate, but instead there is the monosaccharide galactose. The sulfate content in keratin sulfates 1 and 2 is variable, and it may be present on the carbon number 6, whether on the monosaccharide galactose or on the N-acetyl glucose amine. Keratin sulfate 1, it is present mainly in the cornea, in the eye, while the type 2, it is found in loose connective tissue as a part of the proteoglycan aggregate in association with the chondroitin sulfate. This is another figure showing the structure of keratin sulfates 1 and keratin sulfate 2. Regarding dharmatan sulfate, it is another mucopolysaccharide. It consists of repeated disaccharide units. Each disaccharide unit contains N-acetyl galactosamine, and idiuronic acid, but also with variable amount of glucuronic acids. The dharmatan sulfate present in the skin and also in the blood vessels and in the valves of the heart. This is the structure of dharmatan sulfate. This is the idiuronic acid, and this is the N-acetyl galactosamine. Now we discuss the glycoproteins and the proteoglycans. The glycoproteins, they are proteins to which oligosaccharides are attached. Here the oligosaccharides are usually branched. What is important regarding a glycoprotein is that glycoproteins contains more proteins than carbohydrates. What are the functions of glycoproteins? They are represent structural molecules, for example, collagen. They act as receptors on the cell membrane for the sake of binding hormones and also aid in the cellular recognition. Also, glycoproteins act as transporting molecules such as transferrin, ceruloplasmin, and they may function as immunoglobulins for immune function. Also, glycoproteins, they may serve as hormones, for example, thyroid stimulating hormone, the human chorionic gonadotropins, also the gonadotropins, the follicle stimulating hormone, the luteinizing hormone, all are glycoproteins. Also, glycoproteins, they have a role in the coagulation process, for example, the fibrinogen. All these are functions of glycoproteins. On the other hand, proteoglycans, they are proteins that are covalently linked with glycosaminoglycans, and the protein part of proteoglycans are called core proteins. But with the proteoglycans, the unbranched carbohydrate chain, the glycosaminoglycan, may form up 95% of the weight of proteoglycans. In other words, the glycosaminoglycan, the carbohydrate portion, constitute the highest proportion of the proteoglycan molecule. What are the functions of proteoglycans? They are found in most body tissues, mainly in the extracellular matrix, where they are bound to the collagen and elastin, and these molecules, they are negatively charged. Therefore, they bind to cations like sodium and potassium, and therefore may help to attract water to the extracellular matrix. And thank you for listening.